Good afternoon and a very warm welcome to all. So today we are going to discuss about important culture media used in microbiology laboratory. So the culture media which are used in our microbiology, it is a growth medium, or it is the culture media or the growth medium. It is a liquid or gel which is designed to support the growth of the microorganisms. So in the last few classes, we are already familiar with the types of culture medium preparation, etc. So there are the different types of media suitable for the growing different types of cells. Here we can discuss some of the microbiological culture media which are used for growing microbes, especially bacteria or yeast. So now coming to the common media used in routine use. First of all, nutrient broth. It is a general purpose medium which is used for cultivating a broad variety of fastidious and non-fastidious microorganisms. So what do you mean by fastidious? So fastidious means those microorganisms which need certain requirements, special requirements for their growth, such as certain types of enzymes or antibiotics or extra ingredients such as blood, etc. within the media, that are called fastidious bacteria. Then non-fastidious means it doesn't require any special needs, special nutrients. They doesn't require any special nutrients. They are called non-fastidious microorganisms. And so this nutrient broth is common to both fastidious as well as non-fastidious microorganisms with non-exacting nutritional requirements. Now the ingredients. Peptone and yeast extract are the main ingredients coming in the nutrient broth, and this both provide nitrogenous compounds, vitamin B complex, amino acids and other essential growth media. So we here two nutrient broth is seen. One is clear, another one is slightly turbid. This slightly turbid means that there is a presence of microorganism. Clear, which indicates that no growth of microorganism. So here in the method of preparation, it is 500 gram of meat. For example, ox heart is preferred, and it is minced and mixed with 1 liter water. And here 10 gram peptone and 5 gram sodium chloride are also added. Then the pH is adjusted to 7.3. So this is the ingredients and method of preparation. Other uses. As it is used as a basal media for the preparation of other media, and also to study the soluble products of bacteria. So these are all about nutrient broth. Next is nutrient agar. Nutrient agar, it is composed of pancreatic digest of gelatin and the beef extract, which provide organic nitrogen compounds, long chain fatty acids, carbohydrates, vitamins and essential amino acids necessary for the cell growth. So the nutrient agar is composed of pancreatic digest of gelatin, beef extracts, etc. And also agar, the main ingredient in the nutrient agar is agar, and agar is added which helps to solidify the media. And the nutrient agar, it is used for the culture of less fastidious organisms as a general media. So in this nutrient agar, less fastidious, that is the organisms which doesn't require special needs. So such type of bacteria are commonly grown. It is also used for the routine culture of microorganisms from typical environmental samples such as water, food and even air. So it is used as a routine culture of microorganisms from the water, food and even air. And the method of preparation, it is solid at 37 degrees Celsius. 2.5% agar is added in the nutrient broth and thus it can be converted to nutrient agar. How? By heating it to at 100 degrees Celsius to melt the agar and then it slowly gets cooled. And here is the petri plate of the nutrient agar, and here is showing the growth in the nutrient agar caused by different organisms. For example, the growth in the nutrient agar by Klebsiella pneumonia, Morganella morgani, Providentia alcalifaciens, Salmonella typhimurium, etc. So this is the nutrient agar powder. It is the high media, that means the instant media. No need of adding each and every requirements in this media. Already the ready-made media is available for nutrient agar. Nowadays all are using this ready-made media for the preparation of nutrient agar. Next is peptone water. Peptone water, it is most commonly used media because in our laboratory for every test to perform, first of all we are inoculating the required microorganism in the peptone water and then we are doing every further test. So the peptone water is the most commonly used medium. And a microbial growth medium, it composed of peptic digest of animal tissues and sodium chloride. The pH is mainly 7.2 plus or minus 0.2 as the 7.2 plus or minus 0.2 at 25 degrees Celsius. And also this peptone water is rich in an amino acid tryptophan and is used as a non-selective broth medium which can be act as a primary enrichment medium for the growth of bacteria. So it is a non-selective broth medium. The peptone water is used as a growth media and as a base for carbohydrate fermentation media. So for performing the fermentation test, we can preferably use peptone water which helps as a growth media for the required microorganism. And then it is a broth medium used for the detection of indole. So broth medium which is used for the indole detection. And it is the minimal growth medium which is used for determining, also it is a minimal growth medium used for determining carbohydrate fermentation patterns of non-fastidious organism. So in the case of the non-fastidious organism, it is a minimal growth medium used for determining carbohydrate fermentation patterns. And also it consists of peptone 1% and sodium chloride 0.5%. So these are the ingredients, that is peptone 1%, sodium chloride 0.5%. It is used as a base. It is used as a base for the sugar medium and to test the indole formation. So it is used as a base for sugar media and also to test indole production. So this is the picture of peptone water. It can be clear. Next most commonly used medium is blood agar. Blood agar, it is an enriched medium provided with multiple nutrients. So this medium consists of more than one type of nutrients that generally comes as a basal medium for the preparation of blood agar by supplementation with blood. So it is an excellent medium for the cultivation of fastidious bacteria. That means those bacteria which need special requirements such as vitamins, etc. that requires particular nutrients and don't profusely grow in general media. So the organisms which require particular nutrients are usually grown in blood agar and it doesn't grow in the medium like nutrient agar. And in this blood agar, 5% of defibrinated mammalian blood. So the 5% here in the blood agar we are adding defibrinated blood. That means we have removed the fibrinogen content, and it is 5% of defibrinated mammalian blood is used. Also, we use mammalian blood include of human, sheep or horse. Most commonly in the laboratory human blood is preferred. It is quite difficult to get the sheep blood or horse blood. And this mammalian blood is added to the autoclave. So it is added to the autoclave. So now this medium, it is added to autoclaved basal medium to prepare the blood agar medium. So what happens in this condition? That is, after the addition of this defibrinated blood, it is kept in the autoclave for the sterilization, etc. Next is, blood agar is used to grow a wide range of pathogens, particularly those that are difficult to grow, such as Haemophilus influenzae, Streptococcus pneumoniae, and Neisseria species. So here what happens is that it is used for the wide range of pathogens which are difficult to grow. In the normal nutrient agar, these pathogens are difficult to grow. Example for those pathogens are Haemophilus influenzae, Streptococcus pneumoniae, and Neisseria species. But in blood agar they may grow because those organisms are fastidious organisms, and also they require special nutrient requirements. That's why, and that are all available in the blood agar. That's why they are fastidious and they can withstand. So it is also required to detect and differentiate what blood agar is used to differentiate hemolytic bacteria. For example, Streptococcus species. This medium can be made selective also. How? By the addition of antibiotics. So antibiotics, by adding antibiotics we can make this media a selective media. The medium is also differential because blood agar test for the so here also this medium is differential because blood agar test for the production of hemolysin which breaks down the RBCs. So it is differential because it is used for the production of hemolysin which break down RBC. So the RBC in the blood agar is break down by the production of hemolysin which is produced in the bacteria. That's why the lysis or the zone of lysis is seen in the blood agar after the inoculation of particular organisms. So this is the normal blood agar without inoculation which we have prepared. This is the growth of the blood agar. Here you can see a zone of lysis can be seen within the medium. That is because most of the organisms produced hemolysin which lyses the RBC or human blood. That's why the color like this. Next is the method of preparation. I have already discussed. 5 to 10% defibrinated sheep or horse blood is added to the melted agar. So it should not be up to 100 degrees Celsius. First of all, when we are heating the nutrient agar at 100 degrees Celsius, and we are now cooling down up to 45 to 50 degrees Celsius by stirring, and when it reached 45 to 50 degrees Celsius, then we are adding 5 to 10% defibrinated sheep or horse blood. Otherwise, what will happen? It may result in the destruction of the added RBC. So next is